Hi, ladies and gentlemen. Today we're going to be talking about Chapter 22, Speciation. So let's start by defining the term speciation. Speciation is simply the process by which one species splits into two or more species. We call this process over a long period of time divergent evolution or adaptive radiation. And of course, we can go back to Darwin's finches as an excellent example of speciation, where one ancestral finch flew over to the Galapagos Islands. They settled on all the islands. Each island had different environmental pressures. They did not mate for many, many years. And if they ever got back together, they found that they were different species. As a reminder, microevolution are the small changes in allele frequency. Macroevolution are the broad patterns of evolutionary change. So clearly there need to be microevolutionary changes that lead to new species. So this is the first section of the chapter, and biologists, in determining and defining what a species is, they look at morphology, which is body shape, physiology, biochemistry, and DNA sequences when they group animals together. And I've highlighted DNA sequences in red here and put a big red arrow because DNA is really the most compelling evidence to determine relationships and how closely related organisms are. So how then does biology describe a species? The biological concept of a species is when a group of populations that can get together and interbreed and create viable, fertile offspring. And the term viable means the ability to live and survive. So not only are they alive and relatively healthy, but they must be fertile as well, which means they need to be able to have offspring as well. And while organisms that are not the same species can sometimes get together and create a hybrid, that hybrid, however, is unable to bear their own offspring. So when we look at species, we can look at similarities between different species. And your book puts forth these two finches, and you can see, despite the fact that they're different species, they look very similar. And then the article you read from Khan Academy had these two birds that look almost exactly alike, but they are in fact the different species. And then within the same species, we can have great diversity, because if you consider Homo sapien, the species of human, we all look incredibly different. The Khan Academy article took dog breeds, for example, lots and lots of different dog breeds, but they're all the same species and they can all interbreed and create fertile offspring. Now, a cross between a Great Dane and a Chihuahua might be really difficult to overcome, but if the sperm and the egg ever joined and it grew in a animal that could accommodate it, like certainly you wouldn't make the mom be the Chihuahua because the puppies might be really big, but it's certainly feasible. So let's talk about reproductive isolation. This is how species stay separate and actually how they form to begin with. The term reproductive isolation is the existence of some kind of barrier that makes it so two species don't create viable, fertile offspring. Okay, maybe the egg and the sperm never ever get together, or maybe if they do get together, there's a difference in chromosome number such that the hybrid is not able to make its own offspring. Reproductive barriers can be classified whether they act before fertilization or after fertilization. And remember that when the egg meets the sperm, the very first new cell is called a zygote. So we're going to classify these barriers as pre-zygotic, before egg even meets the sperm, or post-zygotic, egg met sperm, but it still isn't working. Your book has a fantastic chart here. I'm going to make it a little bit bigger. Excuse me. Of the different isolating mechanisms. On the left are the pre-zygotic barriers that block fertilization, and on the right are the post-zygotic barriers. So let's start with the pre-zygotic barriers. Let's consider habitat isolation. You have two organisms that live in a similar area. It's possible that they're going to get together and try to do the thing. However, if they don't ever cross paths, sperm and egg are never going to meet. The example your book gives are two species of flies, and one lives on an apple tree and the other lives on a blueberry bush. Those are their preferred niches. And because of that, because they spend most of their time around these specific trees or bushes, they're not going to cross paths and they're not going to share egg and sperm. That would be habitat isolation. They are simply not meeting up at all. Temporal isolation, and the word temporal means timing, means that two species are available to mate at different times. This species of skunk mates in the winter, and this one mates in the fall. They just aren't ready. Timing is off. When one female is in heat, and I'm not sure if that's the appropriate term you would apply to skunks, the male of the species, they may not cross paths, or the female of another species is ready at a different time. So timing can be off, and that is temporal isolation. Behavioral isolation, and I encourage you to click this link on the PDF of the PowerPoint and watch. It's like maybe a 3-minute video of the blue-footed booby, which is just a funny little bird, and their mating dance. And what they do is they pick up their feet and they show their feet and they extend their wings and they bow to each other, and it's very intricate. If the blue-footed booby comes along and shows his feet and spreads its wing and bows to a penguin, you think that's going to get the penguin in the mood? No way. That is a behavioral isolation. So there are certain behaviors that unlock mating for members of their own species, but they do nothing for members of different species. So that would be behavioral isolation. And you can see right here that if they live in the same neighborhood, on the same bushes, and the timing's right and the behavior's right, then, well, they just may get together and try to mate, or what my kids call do the thing. Mechanical isolation is when mechanically the parts just don't line up, or perhaps they don't fit. Okay, I can let you use your imagination there. Gametic isolation is when the gametes don't recognize each other. Okay, so maybe the parts fit, or maybe they're broadcast spreaders and eggs are released and sperm is released in the ocean from sea anemone and corals and crabs all at the full moon. Why does a sea anemone not get fertilized by a crab sperm? Okay, because proteins on the surface of the sperm are not going to be recognized by proteins on the surface of the egg from a different species. That would be gametic isolation. The gametes just don't recognize each other. So even though there was a mating attempt, or at least a spawning attempt for some organisms, it's just not going to happen. Okay, so let's say they live in the same neighborhood, timing's right, the behaviors work, the parts fit, and the gametes actually recognize each other such that a zygote can form. The sperm can actually fertilize the egg. Okay. One of the things that can happen here is the hybrid can have reduced viability. It can start growing in utero or in a shell or whatever scenario, and it can just not grow all the way and fully develop. Or it can grow, be born, and be very weak and die shortly thereafter. That would be reduced hybrid viability. And our last example that we're going to talk about would be reduced hybrid fertility. This is very common with a horse and a donkey when we get them together and we make a mule. Okay, mules are strong workers, and they have some great attributes of both of these organisms, but mules are sterile because horses and donkeys have a different number of chromosomes. So mules have an odd number of chromosomes, and when meiosis happens and animals try to go through making egg and sperm, instead of cutting the chromosome number in half, how do you cut an odd number in half? So no egg or no sperm will have the correct chromosome number, and that's why those organisms are unable to have their fertile, or that's why they're infertile and unable to have their own offspring. Not going to really talk about hybrid breakdown. You can see here that that's plants. Plants really are hard for me because they break so many of the biology rules. And if you want to grow up and be a botanist, I'm sorry if I have insulted you. I say go for it and come back and explain it to me. But this is when there is a hybrid that can actually have an offspring, but those offspring aren't very strong. And we're not going to talk about it because it's really just a plant thing. Okay, a couple more examples of other types of hybrids. This is a growler bear. Okay, it is a polar bear and a grizzly bear hybrid. Okay, it can happen. It's never really going to happen in nature, but because man fools around with things, it happened. This right here on the left is a ligon, and this is a liger. On the left, it's a tigon, and this is a liger. They're both the result of a tiger and a lion, but they're named and they have a little bit of differences based on which is the father. The ligon has a lion as a father. The tigon has a tiger as a father, and the ligon has a lion as the father. My gosh, that's so confusing. Okay. Similarly, we have hinnies and we have hinnies and mules. They are both the result of a horse and a donkey, but a hinny is when the horse is the father and the mule is when the horse is the mother and the donkey's the dad. Okay, that's hard. But long story short, all of these hybrids are sterile. So let's get this a little bit smaller, excuse me. And let's talk about what must take place for speciation to occur. Excuse me. There must be some sort of reproductive isolation. Like we talked about all those barriers, there needs to be a lack of exchange of genetic information for speciation to occur. Okay? Whether they're separated geographically or they're separated within the same geographic area, they are not getting together and sharing gametes for some reason. We're going to define the two ways speciation can occur in these two terms, allopatric and sympatric. Now, I think of allopatric as, see, it says ALLO. I think of all over the place. Okay, they are all over the place geographically. They are separated. One's here, one's here. They are separated geographically. And if you can see, here is your ancestral species. There has been some sort of geographic separation. Well, they're separated now by a mountain range. Obviously, that didn't happen overnight. But because that was such a large barrier, DNA is not being exchanged across the mountain, and two species are going to result. Sympatric, the prefix or the root word sim means same. So sympatric speciation is the development of a new species within the midst of the ancestral species. There was no geographic isolation, but something separated them from sharing gametes, and voila, we have a new species. We'll talk about these more in a little bit more depth here. So allopatric, other country, I prefer all over the place. It just sort of helps me remember it. If you find that annoying, I'm really sorry, but it works for me. And if it works for you, you're welcome. An allopatric speciation, gene flow, exchange of DNA, is interrupted when a population is divided by a geographic barrier. So consider these two different species of fish right here. Millions of years ago, sea levels were higher, and these fish were able to swim back and forth and back and forth and back and forth and share their DNA. Well, about 3.5 million years ago, the sea level dropped, and out from the ocean emerged this land as the sea level dropped. And that land is the Isthmus of Panama. Well, if you're a little fish, you cannot cross the Isthmus of Panama. So suddenly, this population was separated from this population. Over time, due to genetic drift, perhaps, different natural selection, different rates of mutation, the DNA became so different that the two species, should they ever meet again, can't get together and make a fertile offspring. Another example of allopatric speciation would be the speciation that occurred in the Grand Canyon between two species of squirrels. Obviously, the Grand Canyon took millions of years to carve out, but if you're a little squirrel, it really is a pretty significant barrier to try to get from one side to the other and across the Colorado River at the bottom. So it really helped to create that geographic barrier for allopatric speciation to occur. Now, barriers are going to be defined by the ability of a population to cross it. So the Grand Canyon is not going to be a barrier for pollen So plants, that's not going to be a big deal. But for little critters like squirrels, it was a pretty large challenge to overcome. And once again, once the populations are separated, you've got mutation, you have natural selection and differing pressures in each environment. You've got genetic drift that can all change the microevolution of the organisms in the population such that when they get together, they can't create fertile offspring. So lastly, that reproductive isolation is going to be the result of genetic changes and accumulated differences in DNA, so that when they get back together, they don't recognize each other as the same species. They may try to mate, the timing might be right, the environment might be the same, the behavior might be the same, the egg and the sperm actually might even penetrate one another, but ultimately they're not going to be able to create fertile offspring. The other kind of speciation that could occur is sympatric speciation, sim meaning same country. So sympatric happens in the same geographic area. Sympatric speciation is going to be primarily plants. Plants are kind of weird. It happens through a process called polyploidy. When we talk about ploid, we're talking about the number of chromosomes. Haploid is one set of chromosomes. Diploid is two sets of chromosomes, one set from mom, one set from dad. But polyploidy means you have multiple sets of chromosomes. It doesn't work for animals. If we have one extra chromosome, and I'm not even talking about a whole set, we have a condition like Down syndrome. That's just one extra of the number 21 chromosome. It's not a whole set. Animals would never be able to survive if they had a whole set of additional chromosomes. So know that polyploidy is a plant thing, and it's when an extra set of chromosome happens due to an error in cell division. So if you take a look at this diagram, the purpose of meiosis is to cut the chromosome number in half. So this parent cell has 6 chromosomes. The egg and the sperm should have 3 each. Lo and behold, there was an error in cell division, and all of the egg and the sperm have 6 chromosomes. If these egg and sperm get together, they make a new organism with 12 chromosomes, which voila, boom, is a brand new species. So wacko. That would be an autopolyploid. These are multiple sets of the same chromosome from the same genome. An allopolyploid is extra sets of chromosomes that result from 2 different organisms. So here's a parent with a species A with a diploid number of 4, species B with a diploid number of 6. Now, species B divided their gametes just perfectly. It put 3 in each one of them. Species A made an error and put all 4 of them in there. Well, you get 4 chromosomes plus 3 gets together. You've got an organism with 7 chromosomes. So that would be allopolyploid. Don't spend too much time on that. Just know that polyploid means multiple sets of chromosomes, and it's a plant thing. They break the rules, and that's how you can have sympatric speciation. Voila, a new species is going to evolve right in the midst of all the others without any kind of geographic isolation. This is just FYI. It's kind of interesting. At least 5 new plant species has evolved by polyploidy since 1850, and many of our important crops are polyploids. And you can see here, based on this diagram, polyploids can make flowers bigger and fruits bigger. Weird. I don't get it. Habitat differentiation can also lead to sympatric speciation. The example that your book gives about the North American maggot fly, whose original host was the hawthorn tree, some have jumped over to the apple tree, and they are separated in little bitty habitats, and they aren't really sharing DNA. While this isn't exactly sympatric speciation just yet, because they can get together should they meet up and create fertile offspring, scientists believe this is actually leading to a type of sympatric speciation. Sexual selection can also lead to sympatric speciation. I don't really want you to focus on that, but there are different colors of fish in Lake Victoria in Africa that some scientists believe are the result of sexual selection. So in summary, Allopatric versus sympatric. And I'm telling you, you need to study this. These are two terms that are a big fat trap for students to fall in every single year. Geographic isolation, all over the place. There is some sort of major geographic barrier in allopatric speciation that prevents sharing of egg and sperm. In sympatric speciation, there is a reproductive barrier in the middle of the original ancestral species. There is no geographic separation, and it results from polyploidy, which is a plant thing, and of course, natural selection and sexual selection. So the question is, and I can tell you're all thinking it, how long does it take for a new species to occur, and what are the patterns like? Well, there are two different models that generally express how speciation occurs. One is punctuated equilibrium, where there are long periods of stasis or sameness punctuated by quick, rapid change. The gradual model says that there are small, gradual changes over time that lead to new species. Both of these are true and appropriate depending upon environmental conditions. So it really depends on the species and the environment, and each individual instance, whether it's been punctuated equilibrium or gradualism. Okay, we're going to skip that. How many genes have to change before a new species forms? Well, depending on the species in question, it can be one single gene. If that gene controls the way your shell swirls, that can be a major game changer. For snails, coiling of the shells is very important. These three closely related species are shown in image A. In B, it's showing two species whose snail coiling directions are the same, and they can easily mate because their parts match up. In image C, the swirls are different, and as a consequence, their parts don't match up. So if the gene controls the way your shell swirls, which is a tongue twister, it can actually lead to speciation. In flowers, there are a couple of alleles that affect flower color, which can then affect pollinators. And flowers with different alleles at flower color loci, are less likely to interbreed because they're going to attract a different species of pollinators. So it's not going to go from one flower to another and take the pollen with it. It's going to be a whole different species. In sunflowers, it's at least 26 genes that can cause speciation. So really what the answer is, it depends on the organism and the environment in which it lives. So that concludes chapter 22, speciation. Please reach out to me if you have any questions or need any further explanation.